Welcome after a long time. I am Vishwanath from FMCH Baleswar, and today we are going to start a new chapter that is your CNS. What I felt that many of you find CNS very tough, but it is not if you understand the concepts very clearly. And I believe that I can teach you CNS in a never read format, okay, covering each and every silent point that is given in your textbook. Okay, we'll cover some important aspects of CNS and we'll go sequentially. We'll start with the synapses, then we'll go to the receptors and pathways. Pathways will extrapolate the tracts. Okay, when we talk about CNS, generally in sensory CNS and motor also, we have tracts. In the motor CNS especially, we have tracts and ascending tracts and descending tracts. We'll read them in a very simplified way, not in that complicated way which you read in neuroanatomy. And in sensory CNS, we'll talk about pain pathways, analgesic pathways, and all. Okay, and receptors about synapses. So first thing we'll start. This is the CNS lecture one, and in this lecture one, we'll start with the synapses. Okay, we'll start with the very first chapter, that is synapse. Okay. What I will teach is enough for your exams, be it NEET PG or your prof exams or USMLE or PLAB. Okay. So synapses, which you know already, that synapses can be axosomatic, exodendritic. Okay, axon and dendrite, you know, a basic structure of neuron, you know, all of you. Okay. If I want to draw, if you just have to see my drawing skills, then I prefer to draw the dendrites first. These are your dendrites, okay, or dendritic processes. This is the soma or the cell body. This is your cell body or soma. These are your dendrites. To know about all of this, some Nissls granules are present. Nissls granules, which are fragments of rough endoplasmic reticulum plus your ribosomes. And finally, we have a long process which is called as an axon. And we have some coverings of sphingomyelin, that is a myelin sheath. Myelin sheath, you know about myelination, the neuroglial cells for myelination. Okay, I'm not gonna teach all of them in detail now because you have read this part in your nerve muscle physiology already, right? So we'll not go into depth. This is your axon hillock. Okay, and this intervening portions, especially these ones, are your nodes of Ranvier. And finally, we have this type of endings, nerve endings, which you just can simply say nerve endings or synaptic terminal. Many names are there, in fact, or you can say botons, or you can say synaptic knobs. Okay, many such names are there, right? So this is a basic structure of a neuron. Many things are there which I have left, which are not. I'm not going to explain in this chapter because this is CNS. Okay, this is not nerve muscle physiology. So I'll be teaching you what is in nerve, what is the axon hillock, what is the function, what is the trigger zone, and all. We'll read in nerve muscle physiology if I take in future, right? And neurophysiology you can find in my channel. A one shot is also there, so you can read that whole chapter in a very less time, but this is a comprehensive lecture. Okay, so basically we are discussing about synapses, and there are two types of synapses. Synapse, there are basically two types. One is a chemical synapse, another one is a electrical synapse. In chemical synapse, some molecules are involved, some chemicals are involved. That is your neurotransmitters. Okay, neurotransmitters are present here. and in electrical synapse you don't have any neurotransmitter, right? And in chemical synapse the synaptic transmission is in a unidirectional direction. Okay, like for example, here we have one neuron. Let's say this is your dendrites and all. This is your axon, this is your terminal button, and again we have the dendrites. Okay, another neuron. So the impulse conduction will be in a single direction only, right? This impulse conduction will be a unidirectional process, but electrical synapse, basically we have between the cells we have gap junctions. Okay, so let's say here is a cell and here is another cell. So between them we have some channels made up of connexin proteins, and these junctions are called your gap junctions, right? And here the synaptic transmission is multidirectional, right? Or bidirectional, you can say. Bidirectional transmission of impulses. None? Okay. So these are the basic things about the synapses. Okay. And what is the advantage is that when we have a group of interconnected neurons, because it is a multidirectional transport, an important line is there in your Guyton textbook. Okay, this is an important line. Please see. Electrical synapse. Just note down the line. I'm explaining. Electrical synapse promotes synchronous firing of a group of interconnected neurons, right? So basically, what it's trying to say is that when I have a group of neurons, basically in memory functions, if I'm a doctor, then I will have to correlate many things in order to understand what is the probable differential diagnosis or the final diagnosis, right? To reach that diagnosis part, we have to correlate many things are there. What are the signs, what are the symptoms, in which disease it is fitting, and okay. Or if there is a case of Parkinsonism, then what drugs are there for drug-induced Parkinsonism? Is it primary neurodegenerative or secondary? Okay, or it is drug-induced, right? So many things are there. So basically, we have memory functions. We correlate many things. So we have interconnected neurons for that. So if electrical synapses are present, then it will promote the synchronous firing. Like if an impulse is there, then it will be synchronized. It will be synchronized across many neurons, right? So it is basically having a faster conduction than the chemical synapse. In chemical synapse, we have a widespread process. Like you know about the process. Impulse comes, then ACH vesicles will fuse, it will release, the nicotinic receptors are there. Many things are read in nerve muscle physiology. So chemical synapse is a bit time taking, okay? But this electrical synapse is basic thing is that it is very fast. It's fast, right? So this is the main fact, or you can say things which are there you must know, okay? And so chemical synapse, you know about that. We have a presynaptic terminal. We have the neurotransmitter that is released from the presynaptic membrane. Okay, fusion of the vesicle with the presynaptic membrane. Neurotransmitter is released. It acts on the postsynaptic membrane and excite or inhibit or modify. Right. Three signals are there. Either it is a excitatory one. It excites. Okay. So if it excites, we call it EPS. EPS is the excitatory postsynaptic potential. If a neurotransmitter is inhibitory, we have inhibitory neurotransmitters as well, right? Like glycine. So it will show IPS, okay, IPSP, sorry, not ISPP, IPS. Inhibitory postsynaptic potential. Okay, either it excites or inhibits, or you can say modify, right? Or it can modify the potential of the postsynaptic membrane, right? These are the basic things which are there, and you must know these things. These are the basic things which you must have read in the nerve muscle physiology. So synapses chapter is basically an you can say a bridge. Okay, a bridge between your nerve muscle physio and your this part, right? And if you see your Ganong textbook, then it starts directly from the receptors part. So we'll cover the receptors in subsequent classes, right? We'll first know about synapses and what are the properties of a impulse, right? We'll see about the law of power, Bell-Megendi law, and all of them we'll discuss in lectures and all today and future lectures, right? So, basic thing about synapse. Uh, let's let me draw. I will suggest that you also draw with me. It will be better for if your presentation style gets improved a bit. It matters a lot how you present in your exam. So basically, we have receptors here. These are nicotinic receptors for acetylcholine. So these are your postsynaptic receptors. Postsynaptic receptors, right? Postsynaptic receptors. And in the presynaptic membrane, what we have, all this. We have your calcium channels, right? I'm just drawing a basic diagram of how the synapse look. Calcium channel. Calcium channels are there. We have ACH vesicles. Let's say these red ones are your vesicles filled with ACH vesicles, right? Okay. Then we have what are the sausage shaped things? Yes, mitochondria. People ask what is the role of mitochondria? So role of mitochondria is to energy, provide energy for the synthesis. You can write synthesis of ACH vesicles. Okay, so they are basically providing energy for the synthesis of ACH vesicles. So mitochondria have energy producing role, right? And about calcium channels, calcium influx occurs, nerve impulse conduction, you must have read in great detail, right? And about this receptor, another thing is given in your guyton. So I'm following for this chapter, I am referring only guyton, and I'm teaching the things given in the guyton textbook only, okay? Because in Ganon, it gets a little bit complicated if I start teaching about your T snares, B snares and all, it will get complicated, right? So if you see the receptor, this is the postsynaptic membrane. Let's assume this. This is your postsynaptic membrane, and here is your receptor. Let me maintain the color coding. Here's a receptor, right? So receptor has two components. One is a you can say intracellular component. One is a binding component. So it has two components. Let me divide it. This is your which is inside the cell, right? This is your intracellular component. Okay, this is your intracellular component, and which is outside this in which the ligand, what is the ligand here? Acetylcholine. Let's say this is acetylcholine, and acetylcholine is gonna bind to here. Okay, so this is your binding, you can say extracellular component, which is specifically called a binding component, right? You must know these things about the receptor, right? And another thing is about neurexins. So what are neurexins? Neurexins are some proteins which dangle from the membrane. Which membrane? Postsynaptic, presynaptic membrane, sorry. So these green things, this type things, what are these? These are neurexins. You must not have read about neurexins in your nerve muscle physio, because it is given here in a diagram. Neurexins. So what are the neurexins? Neurexins are basically, what is the function is that they are basically proteins, and they are providing structural stability to the synaptic architecture. Okay, they're providing structural stability to synaptic architecture. Okay, so about neurexins you know now, about basic things about synapses you know. Done. Uh, About impulse transmission, you all know that because action potential will depolarize the presynaptic membrane. So action potential depolarizes presynaptic membrane. Okay, sorry. Then what will occur? There will be calcium inflow into the terminals. There will be calcium inflow into the synaptic knob. Here we saw no, there will some calcium channels. So from this calcium channel, what will occur? Your calcium will influx. If you want to know, calcium will cause the fusion of T snares and B snares. Okay, T snares and B snares I have taught in your. I've taken a lecture on nerve muscle physio as well. If you can find in my channel, there I have taught you T snares and B snares in detail. So basically, calcium will cause this. Just know, calcium will cause. There are two type of proteins: T snares and B snares. Okay, so calcium will cause the fusion of T snares and B snares. So it will facilitate the binding of the vesicle, the fusion of the vesicle basically to the presynaptic membrane. This fusion will cause the release of acetylcholine, right? This will open and the neurotransmitter will be released, acetylcholine or any neurotransmitter I am taking, okay? And this neurotransmitter will act on now receptors, right? If it is acetylcholine, then we have nicotinic receptors, nicotinic. We have types of anion channels. We have cation channels. Receptors also are of two types. Okay, so basically, if I want to classify receptors, just write this receptors. So receptors are of two types. One is your ionotropic, one is your metabotropic. One is your ionotropic, one is your metabotropic. You can know from the names. At least this one you can know from the name. Ionotropic means it will directly open your ion gated channels. Like iron, what are the ion gated channels? Your sodium channel, potassium channel, these are your cation gated channels, and chloride channel, it's a bicarbonate channel, these are your anion channels, anion gated channels. So ionotropic receptors are directly opening ion channels, right? They will directly open the ion channels. So we have two types: cation channels and anion channels. Two types, right? So cation channels are the neurotransmitters. Okay, just listen. So I want to tell that the neurotransmitters which open cation channels, that will cause the inflow of positive ions. Okay, the neurotransmitters which open the cation channels are called as excitatory neurotransmitters, and which open, just a minute. So there was a call. Okay, we are learning about, yeah. So the neurotransmitters which open the cation channels are called as the excitatory neurotransmitters, and which open the anionic channels or anion channels, causing the inflow of negative charges, are called as inhibitory neurotransmitters, right? So these are the things which you have to understand and remember, right? This the concepts are clear, right? Okay, these are the things about ionotropic receptors. And metabotropic receptors have a very unique mode of functioning. Okay, they work mainly by a system which is called as your secondary, or second, you can say second messenger system. They work by second messenger system. So second messenger system is basically a type of thing which is promoting prolonged post synaptic neuronal excitation, which is possible, which is impossible by your ion channels. Ion channels are short acting. Okay, they are opening the things and done. Like when a ligand binds, let's say, to have something to show you. Just a minute. Okay, prompt. Okay. So let's say here's the receptor. Okay, this is the receptor, and this is the ligand. Okay, so ionotropic receptors are basically short acting. Like If the ligand binds, okay, excitation is done, go on. Okay, it withdraws, goes back. And metabotropic receptors are acting as second messenger system. So it is facilitating a prolonged and strong action. This is a strong acting receptor. It is stimulated for a prolonged period of time, right? Okay. So this is the concept about second messenger system. And we have to learn here the second messenger system in detail, and that too using the G protein. Okay, using the G protein coupled receptors. The receptors are called G protein coupled receptors, and I will teach you this second messenger system now, because we have this in our chapter, right? In Guyton. Second messenger system. Give the heading second messenger system. Let's learn about second messenger system. So most commonly the second messenger system, there are many types of things like JAK-STAT receptors, your many other. Okay, basically we'll most common type is the G protein using G protein. And how this G protein complex is formed, we'll see. So basically we have an inactive G protein complex first. Inactive G protein complex. So inactive G protein complex is important. Just listen to me very, very carefully. Inactive G protein complex is basically having a GDP. Note the components. GDP. We have a alpha component, which is also called as the activator portion. Alpha component. We have a beta component, and we have a gamma component. So GDP, alpha, beta, and gamma. Right. So basically, what is happening is that when a neurotransmitter is released from the presynaptic membrane, it comes by the synaptic cleft. Do you know the width of synaptic cleft? Have I taught here? This, okay, this width, this is a synaptic gutter or a synaptic cleft. This whole part, just look at this blue shaded portion. Okay, this whole part is your synaptic cleft, and the width of synaptic cleft is 200 to 300 angstrom. This is called the synaptic cleft or gutter or synaptic gutter, right? So basically, this neurotransmitter, when a neurotransmitter is released from the presynaptic membrane, it stimulates the receptors. This receptor will bind. Okay, this receptor, what is the receptor? It is a G protein coupled receptor, right? So this receptor will have a G protein coupled to it, right? So this will open up the binding site for G protein complex, right? So G protein will, this complex will bind to the receptor and will cause the action. So basically, when the receptor is stimulated, it will cause the release of alpha subunit. Okay, this if you are not understanding, let me draw the things. Okay, if I draw it, it will be easy for you. Let's say this is your alpha component. We'll draw it like that. This is your this orange one is your alpha component, and let's say I draw the beta in green and this gamma component in blue, and a GDP will be linked to it, right? GDP, and GDP will have two phosphates. So let me show you the two phosphates like this. This is your GDP, correct? This is inactive or active? This is the inactive complex. Inactive. This is the inactive complex. So what happens is that when the neurotransmitter will bind to the receptor protein, just understand here, let's say take a membrane. I'm explaining the concept given in your Guyton textbook only. I'm not taking any other concept. Let's say this is a receptor. I'm writing R for the receptor. So when the neurotransmitter, let's say this is the neurotransmitter, when the neurotransmitter is getting bound to this part, when the neurotransmitter is getting bound to this part, okay, when it is coming here, this G protein complex will get bind to this one, this whole part. Copy, paste. This whole part will get bind to this. Okay, and when it gets fully bound, okay, So basically, let's say here there was no neurotransmitter. Okay, when there was no neurotransmitter, this inactive complex was bound to it, right? And now the neurotransmitter came and bound to it actually, right? When there is an actual binding. So this is the same receptor I am drawing in the step 2. This is the same receptor. This is the R. This is the neurotransmitter. This is the receptor. And we have the G protein bound to it. Okay, in this part. So when the neurotransmitter binds, this is the step 1. In the step 2, when the neurotransmitter binds, the GTP, okay, the GTP, okay, guanosine triphosphate with 3 phosphates, 1, 2, and 3. This will knock off the GDP out. This will push the GDP. So it is like coming here, and when the neurotransmitter binds, it is pushing the GDP out of the picture and it's getting bound to this alpha subunit. Okay, so basically now how it is looking, how the complex will look when there is a binding of neurotransmitter. This is the alpha complex as it is, this is the beta as it is, this is the gamma, and in the place of GDP we have our GTP. Let's write with red. GTP with 3 phosphates, right? And when there is a binding of GTP, remember, you can just make a trick. Beta and gamma don't like this GTP at all. They like the GDP. Okay, beta and gamma are contraindicated, or you can say they dislike this GTP. When they dislike this GTP, this beta and gamma will leave. Okay, so in the step 3, this beta and gamma leave. This beta and gamma will leave. And this leaving of beta and gamma, this beta and gamma components are separated, and what remains is this part, alpha component with the GTP with 3 phosphate. This is your active G protein complex. This is the active complex. This active complex is the basic thing which will do 4 functions. Okay, it will open up the it can, okay, it has 4 possible functions. Either it can open up the potassium channels. Here we have, let's say, K plus channels. This active complex can open up the potassium channels, can open up K plus channels. First function. Secondly, it can activate some enzymes. Let's say we have a membrane enzyme. I'm writing ME. So what it can do, it can activate, second function, activate membrane enzymes, or it can do third function, that is, it can activate the intracellular enzymes. Let's say we have an intracellular enzyme, ICE. So it can activate, third function, intracellular enzymes. This is the third function. Or it can stimulate your gene transcription. So if we have our DNA here, a new sorry, you can nucleus here. Just a minute. Yeah. So I was telling that I'm a bit feverish. So you might have been seeing that I've been in a depth of that energy or so to teach. Okay. So I'll keep this lecture very short and up to 30 minutes. Like we'll finish up this lecture here. After this, I repeat this concept again. And in the next class, we'll continue with the neurotransmitters and potentials, properties of synapse and all. Okay. So here, so I was telling that if this is the nucleus, this DNA, mRNA, and protein, this is the central dogma, you know. So this GTP, this active complex may activate the transcription. Okay, can stimulate, or the fourth function will be activates transcription of genes, right? So this active G protein complex will have four functions, right? You understood the four functions? Can open up the calcium that K plus channels, may activate the membrane enzymes, can activate intercellular enzymes, and can activate the transcription of genes, right? Yeah. So this is the active complex. You understood what is the thing? Neurotransmitter was there. When there is no neurotransmitter, this inactive complex has been bound to this receptor. When there is a binding of neurotransmitter, this GDP is replaced by GTP, and this beta and gamma will leave. When this beta and gamma will leave and GDP is bound, this is called the active complex. This active complex will cause the actions. What are the actions? It can open up the K plus channels, or it can activate the membrane enzymes, or it can activate the intracellular enzymes, or it can activate the transcription of genes. All the actions are there, right? So any of the action we know can be done. And lastly, last point is that how this G protein is inactivated. This is the activation of G protein. So you can just say that this is the activation of G protein. Lastly, we have a note about inactivation. How it is inactivated? So the active complex, active complex had active complex. What it had? It had a GTP plus your alpha subunit. So this in this active complex, if there is an inactivation to be occurring, then this GTP will hydrolyze. This GTP will hydrolyze, and if it hydrolyzes, GTP is hydrolyzed to GTP hydrolyzes to GDP plus inorganic phosphate. Right? This inorganic phosphate is released, and this alpha complex, alpha that subunit will release the target protein in activating the second messenger system. Okay, so this GDP will be bound to now. Okay, and this GDP is hydrolyzed. This GDP will be bound, and GDP, who loves GDP? Beta and gamma. This will again get bound. So it will again form the inactive complex. So this beta and gamma again combine and form the inactive complex. And this inactive complex, what it had? It had a alpha subunit, beta subunit, gamma subunit, and GDP. Right? So this is the inactivation. Then again, if impulse comes, the neurotransmitter bind, then again it is activated. Again, this whole cycle repeat. Right? So this is in short about your G protein coupled receptors and second messenger system. So today in today's lecture in CNS part 1, we learned about a basic things about the basic structure of a neuron. Then about synapses, we learned about chemical and electrical synapses, and this is fast, and we learned about few important lines. We saw the basic architecture of a synapse. We talked about neurexons is important. This neurexons is important part when can be asked, and we talked about the basic uh, functioning of the receptors, and how the classification of receptors such as ionotropic and metabotropic, and about neurotransmitters, basic things. And we learned in detail about your second messenger system, which is very very important, because it is if you leave this part in physiology, then it will again come in pharmacology. Okay, if you leave this in pharmacology, it will again come in medicine. Okay, so it's ultimately you have to read, right? So second messenger system, and we talked about all of the processes and how the system is working, right? And about the inactivation also. So that completes your part 1 of CNS sensory physio. So we are into your sensory physiology, sensory CNS physiology, and this is the lecture 1. In lecture 2, we'll talk about neurotransmitters. We'll talk about properties of synapse, talk about neurotransmitters, properties of synapse, and few important concepts like occlusion, okay, summation, temporal summation, spatial summation, okay, occlusion, summation, and subliminal fringe. Subliminal fringe, right? We'll talk about these concepts in the subsequent lecture, right? And as always, if you have any doubt, you can text me on uh, Insta at Shanath dot Tripathi underscore, or you can just DM me on WhatsApp on this number. Don't call me, please. Just you can DM me your doubt or query, right? So I hope you enjoyed the lecture. That's all in today's lecture. Thank you and good night.